All right. Thank you, Alice, and thanks for everyone for being here. There's a slight change in the title, but basically it looks like the same hybrid zone looking at the genetic basis and biochemical basis of the introgressing plumage that you saw in the hybrid zone that Kira talked about. So I'm sure since this is a manakin symposium, the audience doesn't need much convincing that the manakins are a sexy bunch of birds. It is a special group of birds that show the effects of strong sexual selection, with the males being able to perform elaborate courtship dances in courts such as the one I'm showing you in the middle. And some species even have special adaptations such as an ability to make non-vocal sounds during performances. Well, of course, the other thing that stands out to us is the colorful plumage that the male manakins possess. Now, this coloration could be caused by carotenoids, melanins, or even structural variation of the feathers. So on the left, I've listed some carotenoid compounds that are responsible to the red or yellow colors that you find in this species. Now, the other thing that's striking in this group is the obvious plumage sexual dichromatism. The female generally tend to look like this, drab or yellowish green. So here the big questions are, how are the different dietary and metabolic pigments get co-opted to produce these plethora of colors that you see here? Well, presumably the whole thing is under the influence of strong sexual selection. And also how the various genes or biochemical pathway evolved during the diversification of the group. So I'm sure we are all well aware of the many recent work based on polymorphic species and artificial crosses to reveal genes that are controlling melanin or carotenoid-based color differences, but not many studies looking at both at the same time. So on your left, I've listed some melanin genes such as the famous NC1R, the agouti signaling protein. On the right, you have carotenoid genes such as the cytochrome P450 and the scavenger receptor genes. Now, to begin to answer questions about color evolution in a group that's under strong sexual selection, and to know what are the pigments involved, we are looking at a study system based on the genus Manicus. So this is a small group with only 4 recognized species. Now, if you look at the map to the right, that's the Manicus or the white-bearded manakin. It's basically just white, gray, and black throughout. And across west of the Andes, you have the golden collar manakin or the Manicus virilinus with its yellow color and the olive green belly. However, if you look at the tree on the right, you see that virilinus is actually sister to Manicus from west of the Andes. So it would appear that this golden coloration is a derived trait. As for the other 2 members, Candida and Orienticus, you can see them here. The basic difference between them is a change in the coloration in the neck region from white to orange in color. So in this study, we'll investigate if the color changes is caused by mere changes in the concentration of one pigment product, or if different species actually have different modified pigment products. We'll also see if melanins are involved in the color changes. Later on in this talk, I will be showing results from biochemistry and microscopy analysis of the feathers. Now, to better understand the precise genetic mechanism underlying the color changes, we will zoom in the hybrid zone that Kira has very nicely talked about in the previous talk. So this hybrid zone is between virilinus and Candida in northwest Panama. Here you have a hybrid zone where the coloration of virilinus, the golden color, and the olive green belly is asymmetrically introgressing into a Candida population. So this bird, the Candida population, is Candida genetically, but looks like virilinus. And the introgression stops at this river, as Kira has talked about. And west of the river, the Candida looks normal. So I'll refer to the birds on the west side of the river as normal or parental Candida, the east side as introgressed Candida. So what we have done is to resequence the genome of 24 birds, 2 from parental Candida A, 10 from both sides of the river, so normal and introgressed Candida, and then 4 parental virilinus from further out to the east. Now, under the microscope for these three species, you see differences in how the feathers look. For Manakin manakin, you can see that for the gray belly, you have melanin being deposited in the barbules. The color is white, so you don't see any coloration, so the feather looks kind of translucent. For the olive green belly of Villinus, you have a combination of the yellow barb and melanin being deposited on the barbules. Now, for the golden color of the Villinus, you have pure yellow color as expected. However, you see a thicker barb and the barbules are missing. So the same goes with the yellow belly of Candii. You have the missing barbules in the yellow belly and the thick barbs, and the neck of Candii looks similar to Manakin manakin, which is kind of translucent. Now, using high-performance liquid chromatography, we found that the orange and yellow color coloration is due only to the presence of lutein. So in this plot, you have the amount of lutein in the feather in the various groups of birds. The Manakin and Candii have no lutein in their color whatsoever. The introgress Candii and Villinus have the same amount, and the highest amount go to Orienticus, and you can see that it has a really deep orangey color in the neck region. Now, moving on to genetic results. So to make sure that we really have plumage traits that are introgressing into a genetic background of Candii, we ran principal component analysis. This PCA plot shows that the Candii populations are clustered together on the left of PC axis 1 and PC 5, which only explain a small amount of genetic variation, separates the two groups of Candii from both sides of the river. So here you have the normal Candii A plus A and B, and then that's on the west side, and on the east side you have the introgress Candii here. Now, let's moving on to some genome scan result. We're using moving windows 10 KB in size to calculate 4 summary statistics across the genome. Any outlier windows are shown in red here. So there are 4 plots. The top 2 plots show introgression statistics, so ABA and relative node depth. A high number for this statistic tells us that the window is experiencing introgression from Villinus into introgress Candii. The bottom 2 plots are differentiation statistics between the 2 Candii populations on both sides of the river. So the 4 statistics show remarkable concordance in showing us which regions might be underlying the introgressing plumage trait. So I'll put in gene names of some of the major outlier regions on top, and then I'll show a table in the next slide. The most prominent among the peaks is this BCO2 over at chromosome 24. Now here's a list of genes I shown in the previous slide. The first 2 are involved in calcium and Wnt signaling pathways. The next 3 genes are involved in melanin transportation, thyroid hormone signaling, or is expressed in melanocyte. And the last 2 are involved with carotenoids, including BCO2, and BCO2 is known to cleave keratin, changing the color from yellow to white. Now, let's take a closer look at BCO2. This is the alignment of SNPs and their genotype in the 24-read sequence birds around the BCO2 gene. Now, you can clearly see the block of introgress BCO2 coming in from Villinus into Candii. Interestingly, among the normal-looking Candii, you also have 2 individuals that are heterozygous with regard to the 2 BCO2 alleles. Now, if you look at the plumage of those 2 heterozygous individuals on the right, you can actually see some yellow coloration being expressed in the neck and back region. So compare that to a bird that's homozygous for the Candii alleles. So we further look into whether any genetic pathway as a whole are enriched for outlier loci versus just looking at individual genes. So for this analysis, we included any genes that are falling within 20 KB of any outlier window. So we found a total of 321 genes. In total, we found 5 pathways that are significantly enriched. The 2 most significant pathways have something to do with angiogenesis or the development of blood vessels. The 3rd pathway, integrin pathway, the integrin protein are involved in the adhesion of cell to extracellular matrix. And then finally, there are 2 pathways that are involved in gut function and the stimulation of gonad activities. So in summary, melanin in the barbules and lutein in the barbs explain color differences in manakins. The largely Candia genomic background of the introgress birds, of the hybrid birds, allow us to precisely locate genes that are responsible for plumage color variation. And because we didn't find any major amino acid changes in the 2 BCO2 alleles, we believe that the changes likely affect cis-regulation of BCO2, and that allows it to be expressed in a sex and tissue-specific manner. And finally, angiogenesis, cell adhesion, and gut function and gonad activation pathway are enriched for outlier loci. So with that, I've finished my talk, and thank you for listening. I'll be happy to take any questions.